Hepatocellular carcinoma is the most frequent primary liver cancer. There are 5 approved treatments for this type of tumor approved by the European and American guidelines. You have resection, you have transplantation, you have local ablation with radiofrequency, you have chemoembolization, these are local regional therapies, and then you have only one systemic therapy that is sorafenib. Aside of these treatments, and after the approval of sorafenib in 2008, there have been more than 10 phase 3 randomized controlled trials assessing new drugs in the treatment of advanced tumors, and unfortunately none of these treatments have shown substantial or clinical benefit for the patients. Recently, there are some studies suggesting some signals of potential benefit, particularly for ramucirumab in second line, and there is a trial ongoing now based on biomarkers, and also new data has come in phase 2 with anti-PD-1 therapies, but we're waiting for the results. Current management of intrahepatic cholangiocarcinoma is based on resection, and if the patients are at more advanced stage, there is a standard of practice of chemotherapy with gemcitabine cisplatin. New data using whole exome sequencing and other types of next generation technologies have identified novel mutations. The most important ones are the fusion of FGF receptor 2. Also, you have mutations in KRAS, mutations in IDH, and mutations in epidermal growth factor and BRAF. So recent studies have shown that around 70% of patients with intrahepatic cholangiocarcinoma may have at least one of these druggable or actionable mutations that are amenable for target therapies. And to my understanding, there are now at least a couple of phase 2 studies ongoing based on trial enrichment for this, for instance, the fusion in FGF receptor 2 and small molecules blocking this receptor, and we will know the results in the early next year. Extrahepatic cholangiocarcinoma, I would say that the understanding of the molecular pathogenesis has not been as explored as with intrahepatic cholangiocarcinoma. We think that these are two different diseases. Extrahepatic cholangio has more mutations in KRAS, for instance, whereas intrahepatic has more mutations in IDH and fusion proteins. So I think that the understanding of the molecular pathogenesis of extrahepatic is still needed to identify novel targets and novel therapies for that. This is the first phase 3 study that, despite that the study is negative, comparing ramucirumab versus placebo in second line, the hazard ratio was 0.86, and there was a non-significant trend favoring ramucirumab. As a result of the subgroup analysis of this trial, the authors identified that patients with high AFP, alpha-fetoprotein above 400 nanograms, these patients may benefit from ramucirumab as opposed to those patients with low AFP. And the hazard ratio for this subgroup of patients is 0.67. As a result of that, the company has launched now another study in second line based on these biomarkers. So we will have a phase 3 study only for patients in second line with high AFP that generally are more aggressive, they have more aggressive tumors, and these patients will be randomized to ramucirumab and placebo, and there is a lot of hope based on the subgroup analysis of the phase 3.